Alright, we're moving on to talk about a special kind of cell division that occurs only in the ovaries and testes, and that process is called meiosis. So before we talk about meiosis, I need to introduce a couple of terms to you so that it makes our discussion easier. The first term I need to discuss with you is the term homologous chromosomes. Homologous chromosomes are pairs of chromosomes that contain the same genes in the same order on them, but they may not be the same version of the same gene. And you get these homologous chromosomes, you get one complete set of chromosomes from your mom and one complete set of chromosomes from your dad. And for example, you might have chromosome number 1 that you got from mom, you've got a chromosome number 1 you got from dad, you've got a chromosome number 2 you got from mom, you've got a chromosome number 2 you got from dad, number 3, etc. from mom, and number 3 from dad, all the way up through 26. You get 26 chromosomes from your mother, 26 from your father. The 26 chromosomes you get from your mom are one complete set of chromosomes. The 26 chromosomes you get from your dad are the other complete set of chromosomes. When I look at the chromosome number 1 that I got from my mom and I compare it to the chromosome number 1 that I got from dad, I will notice that the same genes occur in the same order. In other words, if this is the location for the gene for eye color on mom's chromosome number 1, in the same location on the chromosome number 1 from dad, there will be a gene for eye color. It may be a different version of the gene. Maybe mom's gene for eye color codes for having blue eyes, and dad's gene for eye color codes for having green eyes. But there will be a gene for eye color in that position. And in another position, there might be a gene for whether your hair is straight or curly. In that same position on dad's chromosome, there will be a gene for coding whether your hair is straight or curly. When I have two chromosomes that have the same genes appearing in the same order, that is called a homologous pair of chromosomes. So there's a homologous pair of chromosomes, there's a homologous pair of chromosomes, there's a homologous pair of chromosomes. We only find homologous pairs of chromosomes in diploid cells. Remember, diploid means I have two complete sets of chromosomes. One complete set I received from my mom in the form of the egg. The other complete set of chromosomes I received from dad in the form of a sperm cell. I only see homologous chromosomes in diploid cells because I have to have one chromosome from each parent in order to have a homologous pair. Now, another term that you'll hear me refer to a lot more next week is this term called alleles. Remember I said that mom and dad each have a position on their chromosome for eye color, but the version of eye color that mom has on her gene may be different from the version of the gene for eye color that dad has. The different versions of the same gene are called alleles. So you could have an allele for blue eyes, or you could have the allele for brown eyes, or you could have an allele for green eyes, etc., etc. So even though we have the same genes in the same position on the chromosomes, the versions of the genes we have in those chromosomes may vary between the homologous pairs. This picture, by the way, here shows you stained or dyed chromosomes, human chromosomes, showing you the homologous pairs. It's showing you chromosome 14 in humans, chromosome 15, 16, 20, 21, and 22. And you can see that the staining, it relates to certain regions of the DNA that are similar, and you can see that there's similar staining between the different homologous pairs, indicating that these two chromosomes have the same genes appearing in the same order as and the same is true here for these two genes. They have the same these two chromosomes, they have the same genes appearing in the same order, etc., even though the versions of the genes may be different. I hope that's clear. So, why have meiosis? Okay, meiosis is a process by which we manufacture egg and sperm cells. Egg and sperm cells have to be haploid cells. They can only have one complete set of chromosomes. They are 1N, we would say. The reason for that is you and I, as adults, our cells, our somatic cells, our body cells, are all diploid, meaning they're 2N. Each cell inside has a nucleus, and inside each nucleus there is a complete set of chromosomes that you receive from mom and a complete set of chromosomes that you receive from dad. That makes you have a 2N cell. In order to maintain that diploid number, I need to have an egg cell and a sperm cell that are haploid. The egg cell can only contribute one complete copy of DNA, and the sperm cell can only contribute one complete copy of DNA. If we didn't do that, if let's say we took an individual who was 2N and another individual that was 2N, and we united egg and sperm from those two cells, now we have an individual that's 4N, so it's got double the amount of DNA in the nucleus of each cell. And if they mate with somebody who's also 4N, the next generation, now we have an individual that's 8N. Now this individual has 8 times as much DNA in their cell. If they mate with another 8N person, now we have a person that has 16 complete copies of chromosomes in their cell, and so on and so forth. And over not too many generations, you can literally completely overwhelm the cell with chromosomal material that doesn't need to be there. We only need 2 complete sets of DNA, the one set you got from mom, the one set you got from dad. So to eliminate this problem and make sure that we don't run the risk of doubling the amount of DNA in the cell with every generation, we have a special type of division called meiosis that allows us to generate haploid cells, egg and sperm, that when they unite, restore the diploid state. This process of meiosis has a few kinks in it that make it unusual, but for the most part it's very similar to mitosis. And a lot of times students even confuse the terms mitosis and meiosis. So I'm going to tell you an easy way to remember the difference between the two of them, and it's one my high school biology teacher, Mr. Kenska, told me years ago. He used to say mitosis, mitose, meiosis, my ovaries, which is always sort of funny to hear a guy say my ovaries. But the point being here that mitosis occurs in somatic cells, in places like your toes, your fingers, your head, your stomach, and that meiosis only occurs in the gonads, in either the ovaries or testes. Meiosis is only for the production of sperm and egg. Mitosis, my toes, meiosis, my ovaries. Now there are 2 stages to meiosis. There are 2 rounds of divisions, meiosis 1 and meiosis 2, and in the series of lectures we go through in just a minute, we're going to visit each of these. The end result of meiosis is to produce 4 daughter cells. So here's my original diploid parental cell. Through the course of 3 rounds of division, here's meiosis 1 and then here's meiosis 2, we end up creating 4 haploid daughter cells. That's different from mitosis. Remember, in mitosis, I start out with a diploid cell, and I give rise to 2 diploid daughter cells, each of which are identical to each other and are identical to the parent cell. In meiosis, however, we start out with a diploid parent cell. We end up with 4 daughter cells that become egg or sperm, and each of those 4 daughter cells is haploid. And not only is that different from the parent cell, but each of these 4 daughter cells is genetically different from the other daughter cells that were made. We're going to see how that happens in a minute, but those are the key distinctions between mitosis and meiosis. All right, let's talk about why it is that daughter cells are not identical. During meiosis, particularly during meiosis 1, the first round of division in meiosis, we have a couple of different unique events which occur that mix up and shuffle up the DNA and the chromosomes so that we get increased diversity in the cells that we eventually create. The 2 ways in which we increase genetic diversity in daughter cells is through an event called crossing over, and then in a second event called independent assortment. I'm going to tell you right now, I will take you through the step-by-step with me talking. There are some really good animations, though, available to you online that will assist you in understanding this. Sometimes it helps to just see the chromosomes moving around and swapping DNA. It makes it make more sense than seeing static pictures like what I'm showing you. So keep that in mind and do take advantage of those animations when you have a moment. During crossing over, this happens during the first prophase of meiosis 1. And what happens is the homologous pairs find each other. So chromosome number 1 that came originally from the mom and chromosome number 1 that originally came from the dad find each other, and they literally physically connect to each other at varying points along their length. This is called synapsing, when we physically connect with each other. And they line up their genes very, very carefully so that they are in exact alignment. This creates something called a bivalent. Okay, it's like a double chromosome. Now that we have this bivalent formed, we then swap material. Okay, let's say the gene for eye color is housed here on mom's and housed here on dad's. We might swap them out. So now a little bit of mom's gene in red is on dad's gene, and a little bit of dad's gene is now on mom's gene. So we literally swap out some pieces and parts between the two homologous chromosomes. This is called crossing over, and it helps to increase genetic diversity, and it explains why you can have brothers or sisters and have the same mom and same dad and yet look very different from each other. The second event that occurs, oh, and by the way, I need to highlight this. This event that I'm talking about right now is happening inside, say, a precursor egg cell. So when I say the chromosome from mom and the chromosome from dad are swapping material, this is inside your mom's egg cell. So it means the chromosomal material she got from her mom, who is your grandma, is swapping with the chromosomal material she got from her dad, who is your grandpa. Okay. The next thing that happens during metaphase 1, which is also part of meiosis 1, is an event called independent assortment. And you'll remember that in mitosis, the whole point of mitosis is the chromosomes line up along the metaphase plate in preparation for moving to one pole or the other during anaphase. In meiosis 1, in metaphase 1, of meiosis 1, the homologous pairs line up as pairs along the metaphase plate. But how they line up can vary. You can have the chromosome from grandma lining up on chromosome number 1 from grandma lining up on the left, and chromosome number 2 from number 1 from grandpa lining up on the right, or maybe you get the reverse. And that's true for each of the 26 chromosomes. It's completely random whether the chromosome that came from the maternal parent is on one side and the chromosome from the paternal parent is on the other side. Totally random. This is called independent assortment, and it further mixes the DNA that grandma and grandma contributed to making your mom. Okay, last slide here, and then we can take a break. Actually, we got 2 more slides, I'm sorry. And then we can take a break before we move on to meiosis 1. We are talking about events that are necessary for the manufacture of sex cells, of what are called gametes, egg and sperm. Sometimes people wonder why we have sexual reproduction in the animal kingdom in the first place. And the answer to that is that sexual reproduction greatly increases the amount of genetic diversity or variation in a population. And this is really important for things like disease resistance. If everybody in the United States was genetically identical, it would only take one deadly virus or one deadly bacterial infection to kill out the entire species. By having us all have different genetic makeups, we have the ability for some of the individuals in the population to be able to persist in the face of a disease and survive to pass genes on and thus ensure that the species continues even if certain individuals may die off. So creating gametes is the ways by which we create, or the cells by which we undergo sexual reproduction. And you remember what I talked about before, the purpose of meiosis is to make sure we create those haploid cells so that when 2 haploid cells, an egg and a sperm cell, join together, they restore the diploid state, and so that we don't keep getting a doubling of the chromosomal number inside the nucleus with every time sexual reproduction occurs. The other event that helps to increase genetic variation, remember we already said that just in the creation of an individual, either egg or sperm cell, we have these events of crossing over and independent assortment that help to increase diversity. Well, we also increase diversity when the sperm and egg cell are allowed to unite together to form the diploid zygote. And that explains why, for example, as I said before, you can have the same mom and dad and you and your siblings look very different. This picture out of your book's a great one. Here's mom and dad, and if you look at their 4 offspring puppies, none of the 2 puppies are exactly identical. They all have slightly different markings. The reason for that is there was a different egg and a different sperm cell that united to create each of those puppies, and with each of those different egg and sperm cells, there were different crossing over and independent assortment events that occurred that led to the overall variation between the siblings.